Slides okay? Video?
Yes.
Okay.
Yes, ma'am.
Thank you so much, Abby. Okay. So, good morning again. It is my pleasure to host this MSI at 50 seminar today. And again, I am honored to introduce the speaker. So, our speaker for today is the head of the Protein Biochemistry and Biotechnology Laboratory here at the UPMSI. PBBL, so you can just refer to it as Pebble. Dr. Eizadora Yu is currently a professor here at MSI and also serving as our associate dean for mentoring and something. So ADMAPA. But the most important part there is the mentoring part. Okay. And prior to serving as our ADMAPA, she was also our ADSAPA and served as an associate professor at the Institute of Chemistry until she joined MSI in 2020, right before the pandemic. Prior to the Institute of Chemistry, Dr. Yu earned a bachelor's in chemistry degree from UP Diliman. And during that time, she also got her master's degree from the Institute of Chemistry in UP Diliman and served as an RA here at UPMSI with Dr. Annette Binez. Her MS thesis advisor is Dr. Virginia Monye. Afterwards, she left for the US to pursue a PhD in biochemistry and molecular biology at the University of Maryland in Baltimore. After earning her PhD, she served as a researcher at the Sandia National Laboratory in the US, where she honed more of her specialization in mass spectrometry. Now, we are harnessing all of the benefits of the years of experience of Dr. Yu in mass spec. So if you are ever interested to work in those fields, you know who to contact. We have Dr. Yu right here. Before I give the floor to Dr. Yu, I just would like to share with you a personal thing. When I joined MSI as an RA and I decided to pursue a master's in chemistry, having been graduated from UPLB, I did not know which subjects to take for my master's, so I asked Aiza what subjects to take, and I asked her, should I take Chem 211 and Chem 250 at the same time in the same semester? So, just for context, 211 is advanced inorganic chemistry, and Chem 250 is physical chemistry, specifically quantum chemistry. Those are the two worst courses you can ever take at the same time. As you just said, kaya naman. And so I did. At yeah, by that time, semester was like, Dr. Yu, well, Aiza during that time is on a totally different league. So a league of her own. Without further ado, I give you from the phone as our speaker today, Dr. Eizadora to you.
Thank you, Eileen. I think hindi na talaga ako mapapatawad na in-advise ko siyang pagsabayan ng in-org at saka FICAM. Okay naman. Ganun. Sige. All right. So, thank you so much for being here. It's my turn to give a lecture for MSI. And I'm kind of excited kasi medyo marami kaming, yung mga estudyante ko maraming outputs ngayon. So, I'm really happy to share what they've been doing for the past 3 years. Taking note na lumipat kami sa MSI during the pandemic. So, naglipat kami ng lab. Okay, so I have a very basic outline of what I want to talk about today. So, essentially, I just want to share with you my journey and why we want to do this and why I choose mass spectrometry, right? And then we'll go on and talk about 2 topics that we have been working on in the lab yung shipworm symbiont, Darwinobacter turneri, and here our favorite sea cucumber. We're going to make sea cucumber sexy after this. And then conclusions and outlook. Okay. So I want to start with this slide because when I was a graduate student, doon pa lang lumalabas yung papers ni Roger Chen about GFP. So when I chose the field that I want, I really wanted biochem because it is the chemistry of life and it seeks to explain life at the molecular level. Yung palaging, bakit? Why is the sky blue? Bakit? So bakit ganyan sila mag-act? Bakit ganito? So fundamentally natin molecular mechanisms. And so I show this slide all the time because if you look at this, there's a jellyfish, Aquaria Victoria, it's bioluminescent, and it all started when Shimomura discovered the protein that actually made it fluoresce. Yung bioluminescence. That was 1960s, 1970s, before I was born. But after that, just the extent was jellyfish. A lot of people work on the biochemistry. Martin Shelfie, they're able to solve the structure of the GFP, green fluorescent protein, which is this barrel-shaped protein. There's a chromophore inside. And that's the one that's responsible for the jellyfish to grow, so GFP, right? And then, naman, katapos na malaman how it works, right? Si Martin Shelfie and his team were able to now recombinantly express this protein in bacteria. So that was revolutionary as well because now you can mutate, right? Sorry, you can mutate the protein, you can change its properties, and you can, make this fluorescent protein paint box. So essentially, yun yung ginawa nila. So sila Roger Chen, it was a 1995 paper. I just entered grad school and I was so excited about this, that we can actually mutate proteins and then come up with something different. And so as you can see, the rest is history. For anybody who's doing molecular biology, ito yung ginagamit natin for imaging, right? So anytime you want to image cells, you use the GFP as the fluorescent protein as a marker. It lights up. It can light up different parts of the cell, neurons, etc. And so, yan yung aking spark, right? And then from the time it was published, right? So I guess 1990s na commercialized yung fluorescent proteins. 2008 they won the Nobel Prize for chemistry because of this. So 1960s, 1979 pinpublish ni Shimomura yung discovery. 20 years to get to. This is a very good success story. Okay. And so basically when I was thinking about what do I want to do with the rest of my life as a researcher, I really want to look at the biomolecules, the molecules that make up our marine organisms, because they have very interesting adaptations, especially lalo na ngayon with climate change, how do these biomolecules, talk to each other and adapt, right? And make sure that the organism lives. And besides that, so yun yung ecology research, sustainable use, conservation, but we can also use these biomolecules for health and other applications. And so a lot of my biotech colleagues are working on drug discovery. Lilibet's working on drug discovery. Si Irene is also working on nutraceuticals and pigments. And the biotech professors that came before us, ito talaga yung kanilang focus. So sila Coke, sila Ma'am Giselle, si Ma'am Luli, di ba? National Scientist Luli. So essentially, this is what we want to come up with. So kind of like a framework where you can study all the biomolecules and then look at the biology and ecology. So for curiosity and also for basic research, but at the same time, try to figure out how we can make use of these for our purposes. Okay, so this is a simplified marine biotechnology pipeline. Very simply put, you have the discovery, ano meron, and then once you discover them, how do you develop and design those things to become into products? And the timelines are different, no? It depends on what kind of products you want to make. But from the discovery perspective, parang kulang pa tayo. So dito dapat talaga tayo mag-focus kasi marami pa tayong hindi alam. So therefore, we have to, be part of biodiversity studies, sequencing the genomes of these marine animals, and from my end, compound identification. Okay. And so once we know what's there, we can figure out, okay, how can we now make them into products and develop lab scale applications, increase the yield, change the processes. So ito mga chemistry and engineering solutions na. And then ultimately, the idea is sana down the line, merong biotech company na sa Pilipinas na merong magla-license ng technology para mapakinabangan natin. So maraming tools of the trade yung biochemists. I think what I just want to focus on is, the main bread and butter of our lab, which is mass spectrometry. Essentially, when I talk to people, sabi ko, weighing scale kami, di ba? If you look at each chemical compound, di ba, meron siyang molecular formula. And the molecular weight is very specific for that compound. So basically, you can identify compounds by just looking at their molecular weight, right? And then I love this technique because it's very versatile. It doesn't care whether you're looking at small molecules, metabolites, proteins, lipids, even DNA, right? Nilad ko muna yung glycan kasi hindi pa namin siya nade-develop. Pero in principle, any compound that has a mass that you can charge, you can actually analyze by mass spec. So ito yung beauty ng technique na to. And by doing mass spectrometry, by weighing the molecules, right, you can figure out whether it's a sugar, it's a lipid, it's ATP, etc. But it doesn't end there because some molecules have the same mass, diba? Isobaric. So meron kami ginagawang technique called tandem mass spectrometry where we actually pulse in gas. So parang carefully we put them inside the instrument, and then once they're inside the instrument, we break them apart. So para sa mga estudyante mahilig sa puzzles, eto na yon. So once you break them apart, now how do you put them all together back, into something that makes sense, that everything is like neat, diba? Pag nag-jigsaw puzzle kayo, yung walang piece na matitira. So yun yung ginagawa namin. So, and by doing that, you can definitely identify compounds. Anything. And so after we do the spectra, we can identify compounds, like I said. For the most part, dati mano-mano siya, nung estudyante ako. So you need to learn a lot of organic chemistry to push your electrons and figure out where the bond, which bond was broken. Pero ngayon, marami ng database. So you can now search spectral databases, you can search sequence databases, molecular databases to be able to, get compound identification. So thankfully, the UST, when I came back in almost 10 years ago, they actually invested in this high-tech instrumentation. So yung DDHP program, they were able to buy this high-end LCESI QTOF quadruple time of light mass spectrometer. So it's really one of the high-end mass specs. And so a lot of almost all of the things I'll show you were obtained using this instrument. Okay. Okay, so yung first story ko yung pteridinobacter ternary enzymes. So essentially the impetus for this is that there's a huge industrial enzyme market, globally. And if you look at the enzymes in the market, karamihan dyan ay household, diba? Yung mga ginagamit nyo sa detergent, diba? When you want to clean your clothes, you have lipases, you have proteases to actually remove the stains. And those are added to the detergent. So a lot of it is household care. And then you have some portion that's looking into feeds, diba? Yung how to deconstruct the feeds for animals. Kasi yung monogastric animals. And then there's also biopharma. Okay, so medyo malaki yung industrial enzyme market. But unfortunately, like like other companies, walang biotech company sa Pilipinas. Walang enzyme biotech company sa Pilipinas. So lahat ng enzymes na ginagamit natin ay imported. Even the enzymes we use for our research is imported, right? Yung taq polymerase, etc. So there's huge potential if we can make our own enzymes. Okay. And for the most part, when we look at enzymes for processing, we want to look at how you can deconstruct agricultural and shell waste and use them as precursors for many things, diba? Other things. So valorization of waste. So you can figure out, for example, yung lignocellulose, pwede mo i-degrade para maging feeds ng mga pigs and chickens. They can also be precursors for biofuels, diba? And also fine chemicals. And on this side, I put detoxification because there's also a lot of enzymes that actually are used to detoxify chemicals. Okay? So, nakita niyo sa side, nandun nakalagay yung iba-ibang enzymes na pwede nating itap. So, a lot of them are oxidoreductases, and then you also have your glycosylases. Okay? And which brings me to this one. So, buti na lang hindi pa tayo magla-lunch. So, I was tapped by Lilibeth. So basically, there was a big project, PMS ICBG, with Margot Haygood and Gisela Concepcion a few years ago, and that morphed into we had secured follow-on funding for the Tamilok program. So essentially, the idea is, if you see this worm here, it's a shipworm. It's actually a mollusk, and it buries into the wood. So yung buong life cycle niya nandun siya sa loob ng wood. Tapos paano siya lumalaki ng super gigantic worm? Kinukuha niya yung cellulose, yung sugars from the wood, at yun yung ginagamit niya as energy material. And just to cut the story short, the shipworm can do this because there are bacteria in the gut of the shipworm that can actually process this cellulose. So gut microbiome. And one of them is Pteridibacter turneri, which if you look at the genome of this guy, it's like half of it is carbohydrate active enzyme. So you can just imagine, di ba, kung ang genome mo kalahati ay para gumawa ng carbohydrate ng celluloses. Ibig sabihin, malaki yung importansya nun sa buhay ng okay. So what I'm going to talk about is some of the research that we've been doing at Pebble. So ito yung mga estudyante ko na nag-work out the pandemic. Actually, I want to shout out sa kanila kasi a lot of these were done at the height of the pandemic. It's very hard to culture bacteria only to say na, okay, shut down ulit. Tapos, okay, tulog mo na yung mga bacteria and then i-reanimate mo sila. So it's very difficult what they've done. But they've managed beautifully. So for the most part, I want to talk about, what we've been doing with Pteridibacter. So number one is genome mining. Essentially, you want to look at enzymes. Like I said, the genome is there's a lot of enzymes there for carbohydrate degradation. So we want to look for enzymes for these, hemicellulose, pectin degradation, and bacterial biofilms degradation. And then we try to attempt, recombinant protein expression. Ito talaga yung gusto namin gawin. And then I'll tell you yung pitfalls and yung mga frustrations namin, but no, we're not giving up. It's just setback. And then the last story would be the proteomic identification of the secreted enzymes. So Renz, Lyle are my MS Chem students, and then Joan and Aidan are MS Marine Science students. Okay, so at the heart of it, what we really want to come up with is an enzyme cocktail for biomass degradation, diba? Paano tayo gagawa ng cocktail na pwede mo siyang i-expose sa abaca or sa, rice hull or sa coconut, and then deconstruct that into simple sugars. And so there's two ways. There's two ways to skin a cat. There's two ways to do this. Number one is if you know the genome, right, of your bacteria, you can mine. You can mine the genome. You can figure out, okay, let's choose which genes to express, and then let's test them. So that's the first one. We synthetically produce an enzyme mixture so that we can optimize the ratios of enzymes, and therefore we can get that. Now, the other way to do this is to figure out what the bacteria produces, diba? Kasi Mother Nature's already perfected this. So we also want to figure out, okay, so anong enzymes ang pinoproduce ni bacteria to be able to do this? Okay, so then for this, we want to identify them by mass spectrometry. Okay, so in strategy one, kasi nga maraming ng genome available, so we looked at the genomes of the Pteridinibacter. We did a lot of analysis on the genes, on how similar or dissimilar they are, and then we chose our candidates, okay? And then from the candidates, we then did some molecular biology where we incorporated the gene into a vector and then tried to have E. coli make those genes or those enzymes. And for the most part, we were good, right? So we were able to produce them, we were able to clone, we were able to sequence, we were able to produce the proteins. But medyo hit and miss kami dito. Sometimes we have active enzymes, sometimes we don't. So it's really just the nature of the game. Okay. So I think we have 25, right, or more plasmids for this, and then we have some enzymes already purified. And so when we have the purified enzyme, the first thing we need to do is to figure out how much is the yield of the enzyme, and then number two is how active is the enzyme. So kinakarakterize ng mga students ko yung activity ng enzymes, yung enzyme activity. And then, tsaka na namin siya paglalaruan. So yung isang gusto namin gawin is abaca fiber softening. Remember, abaca very rough. Paano siya gagawin for textiles, for clothing. And so what Joan did was to immerse the abaca fibers in enzymes, thinking na baka pwede siyang lumambot. And this is an SEM picture where you can see that this is the untreated, medyo magalasgas, tama ba yun? Rough. And then after a few hours of exposure to the enzyme, lumalambot siya. So we're kind of excited kasi, marami kasing gustong gawing textile, yung fiber, yung abaca. And then the other thing we looked at is bacterial biofilm degradation. So if you notice yung bacterial infections, they form biofilms. And the biofilms are ectopolysaccharides. And because they're polysaccharides, pwede silang ma-degrade ng enzymes na pinoproduce namin. And so what Joan did here was to figure out if we can disrupt biofilm formation or actually disrupt the biofilm that's already there. And I know it's a busy slide, but it's kind of promising kasi yung data na nakita namin is that pag nilagyan mo na siya ng coating ng enzyme, almost 50% yung disruption ng formation ng biofilm. So it could be used for antibacterial, the devices. You want to coat them para hindi sila magkaroon ng bacterial biofilm. Okay? Okay, so ginagawa namin to, we came across a lot of problems actually in doing recombinant protein enzyme technology. Sa ibang bansa kasi may robots, you can make like hundreds of the enzymes, and then you do yoga and then you come back, and then when you come back you already have your enzymes, diba? Tapos pag nagkamali or hindi siya nag-grow or hindi siya maganda yung sequence, mag-order ka lang. So medyo mahirap maging ma-adapt siya to Philippine setting. Yung bilis yung pag-set up ng recombinant protein expression. Lalo na kung express mo something that, is not there before. And so I think for this, what we're trying to do now is to collaborate with MBB, sila Rondi, sila Pia, to be able to make sure that, what we do is actually adaptable to the Philippines. Pero we did create plasmids, we did create enzymes. So it's just a matter of figuring out, we ran out of funding. So it's just a matter of figuring out where to put them or where they can be used. All right. Now, the second strategy, kasi nga sabi namin, parang hit or miss you choose the candidate genes from the genome. What if there's hundreds of these genes, so how do I choose? So the next strategy is to look at what nature makes. So we have the Ternerae, the bacteria that we isolated, and then pinapakain namin sila. So we give them in different carbon sources, cellulose, silane, and pectin. These are all different plant cell wall polysaccharides. And let's see kung mag-grow sila. So lo and behold, excuse me, they grew naman, diba? So ibig sabihin, they were able to utilize the carbohydrate. And then because they did that, the next thing that we did was to figure out, okay, so aling enzymes are responsible for actually degrading the plant cell wall. And for this, we did proteomics, no? MS analysis and protein identification. Okay. Cute yung story nila Lyle at nila Renz. Kasi if you look at the secretome, so E7 is one of the isolates that we were working on. It's from Mabini, Batangas. And pag tinignan mo siya, so ang ginawa namin, meron kaming bacteria that we grew in different carbon sources. Like I said, one is in sigma cell, which is cellulose, one is in cyllan, and one is in pectin. And then we harvested that. Tapos pagka-harvest namin, chinek namin yung activity ng secreted enzymes. And so what was interesting was yung sigma cell and na cocktail was able to degrade both cellulose and cyllan. Okay? But the cyllan secretome can only degrade cyllan. Hindi niya kayang kainin si cellulose. So, ano siya, specialist, di ba? And then yung pectin secretome, parang nagulat siya kasi 'di ba sa in nature wala namang pectin yung shipworm. So I guess nagulat siya, ano kaya tong pinapakain sa akin? And so it's trying to throw everything at the polysaccharide. And so for pectin secretomes, they were able to they were active against cyllan, cellulose, and pectin. Okay? And nakikita rin namin na tumataas din yung protein content. So these are good. So, ano yung mga proteins na yun? So, basically, they analyze all the proteins in the cocktail, and it's kind of a busy slide, but the take-home message is this bacteria, produces a common core or set of proteins for plant cell wall polysaccharide degradation. So if you look at this, typically they have like, 1/4 of the enzymes as a carbohydrate active enzymes. There's also non-casein enzymes. There are other proteins. And then if you look at this slide over this graph over here, ito yung parang make-up ng secretome. So if you have sigma cell, a lot of it is carbohydrate active enzymes, right? But look at pectin. Pectin produces a lot more, ancillary enzymes, non-casein, other proteins. So there's something there. And then when we look at the intersections, of these core, so there's a set of 8 that's always present in the secretome. So we're kind of excited kasi we know what they are. I'm not gonna show it kasi malay natin, in the joke, for ID, for purposes. But we have identified 8 enzymes that can be used, and then the next step would now be to generate them, recombinantly express them so that we can figure out how to use them. Okay. Now, this is an offshoot of the proteomics study that we did. So in identifying namin what is there, in identifying ng mga students, and then they try to figure out protein interaction networks between the enzymes in the cocktail. And what was interesting was that the enzymes are sort of working synergistically, and they can only work because they are predicted to interact with the rest of the protein via this protein. So we're trying to figure out what this protein is and express this, kasi why is it always at the heart of the network? Importante siya, 'di ba? So ano kayang role niya dun sa degradation? Okay? And so for the shipworm story that we have, so based on proteomics, what we've figured out is that the T-ternerate secretomes are largely similar, and it suggests that they secrete a protein set of enzymes that is central to degradation, so they always produce this. Based on this list of proteins, we were able to predict interactions and functional associations, and this implies synergistic and cooperative action among the proteins. So, importante siya when we're going to make the cocktails, okay? And hopefully, we will find money and then figure out how we can actually express and characterize these enzymes in the future. Okay? All right. So yun yung enzyme story namin. Then I want to switch gears and talk about sea cucumbers. So medyo interesting din yung story ng sea cucumbers. Now, the sea cucumber work, it started because Mom Annette was talking to me about a lot of things she wanted to do. Diba? Parang marami kasing behavior si sea cucumber. Tapos she was interested in how it actually does that chemically. Molecularly, can we assay? And so, I'm scratching my head, like, how do we do this? The other thing was also she was trying to figure out if they can have chemotaxonomic markers kasi may cryptic species eh. So yun, maraming pag-uusap dyan sa lobby. Maraming mga notebook na items na ito yung dapat kong isipin. But of course, since wala tayo alam sa kanya, the first thing to do is figure out what's there. Okay? And the sea cucumbers, it's really, I want to point out that the sea cucumbers are actually a very rich source of bioactive compounds and additives for several industries. So in Malaysia and Korea, the sea cucumbers extracts are used as ingredients for personal care products. We've seen the face masks with sea cucumber essence, right? Whatever. And then you've also seen personal care products and even health tonics. Sa Malaysia, merong very prolific gamat industry. So gamat is their sea cucumber extract, and so they have this ointment that you can use when you have wounds para gumaling siya. At yun ay sea cucumber. So these are all very established industries outside the Philippines, right? So and lately sa atin sila kumukuha ng raw material kasi na-deplete na yung kanilang resources. So in a way for us, it presents an opportunity, right? So if we can create our own industries from our own, resources, that will be good. And so, but before we can do that, we need to figure out what's there. How do we now make use of this? And these are expensive. So, if you look at the sea cucumber capsules from Amazon, it's like $250 per bote. So there's an opportunity there. And if you look at the sea cucumber literature, ang dami nang naaral sa sea cucumber, no? So if you look at this, it's a lot of compounds. There's amino acids and peptides. There's collagen. There's saponins. There's sterols, fatty acids, etc. And iba-iba yung activity nila. There's a lot about wound healing, tissue regeneration, anti-cancer, anti-proliferative, antimicrobials, immunostimulatory, and then also for heart health. So marami siya, 'di ba? And in fact, parang yung functional food na sea cucumber nakukuha din dito. Okay? And so if you look at the literature on sea cucumber natural products, this is just a PubMed search of the papers, with sea cucumber compounds. Ang dami na from the 1960s. So parang may boom na yon, maraming pag-aaral. Pero the green ones actually is the metabolomics. Ito pa lang, konti pa lang yung nag-aaral ng mass spectrometry, metabolomics, proteomics of sea cucumbers. And a lot of these are on Apostichopus japonicus, which is, what the Japanese are actually interested in. So wala pa on Stichopus horrens. Okay. So, which brings me to my next point. So, how do we do natural product discovery? There's two ways. There's a traditional way, the bioassay guided targeted approach, wherein you have a bioactivity in mind, and then you screen for that activity, and then you identify what is responsible. Ito yung ginagawa natin palagi. But I also want to put forth a complementary approach, right? Which is the omics based approach, which is untargeted. When we come up with these extracts or these organisms, essentially what we want to do is to come up with a comprehensive list. Walang bias kung ano yung meron doon o para saan siya. Gusto lang namin malaman ano yung meron yung animal. And then once we have the comprehensive list, we can identify targets. Remember, a chemical compound is the same whether you get it from sea cucumber or seaweed, if it's already reported to have some activity, diba? Then you could also say na, okay, that particular compound with this structure will also act similarly. So, ganun yung idea. And so we can, once you identify the targets, you can now figure out, ito maganda para dito, ito maganda para dyan. And in a way, you can also figure out novel or new compounds, diba? Kasi hindi pa siya na-match diba? And we do a lot of searching based on clues from biology, because a lot of these bioactive compounds are actually adaptations of the animals. So in the case of sea cucumber, marami siyang anti-predatory mechanisms. So ang saponins, deterrent siya. So sa ecology yun yung kanyang purpose. Pero once we now get the saponins, this could be anti-cancer agents, this could be something else. So we really need to look at biology to figure out, ano kayang possible compounds that are there. So this is Stichopus horrens. It has unique anti-predatory mechanism. So ang tawag sa kanya ay hanginan. Kasi pag tinaas mo siya sa tubig, pag nahanginan siya, either nalulusaw siya, like this one, or naninigas or lumalabot. So depending on, the stimulus. So there's a lot of activity there. It can slough off the dermis. So bawa, kung gusto mo siyang inhibible, yung sea cucumber, we've seen where sometimes the sea cucumber, like sloughs off the skin. Sige, sa'yo na yan. Aalis na ako. So it sloughs off the skin and it regenerates. Right? And then this one is very drastic, body liquefaction. Pag nag-liquefy na, wala na, dead na yung animal. So, di ba, isipin mo, what could possibly be the biochemical trigger for an animal to think na, okay, I'll rather die than be touched by this fish or whatever. So may mga ganung adaptations. Okay? And it's the raw material for gabat in Malaysia. So what I want to show you in the next few slides is our preliminary work on the small molecules and collagen from Stichopus horrens. Okay, and so the work that I'm going to show is from this talented pool of students. So Suzanne, Ralph, and Kim are MS Chem students. And Danielle and Aiden are MS Marine Science students. So I'll talk first about our data on global metabolome profiling, and then segue into yung proteomics and collagen characterization for biomaterials. So a lot of this is in collaboration with my friends at chemistry, yung mga organic chemists doon, tsaka physical chemists. Okay, so for the metabolomic profiling, this is the instrument that we use. It's the QTOF mass spectrometer in chem, and essentially the idea is, you extract everything that you can from the tissue. So we use the body wall, the viscera, we use aqueous or ethanolic, and essentially try to identify everything that's there. And we do it both in positive and negative mode mass spectrometry, kasi some compounds can be charged positively, some are negative. So para lang comprehensive siya. And then we did some colorimetric quantitation for some segments, some classes of compounds. Okay. Okay. Mahirap mag-mass spec ng metabolome. Kasi the sequences are different, the structures are different, and so therefore, if you think about breaking the bonds, there are so many ways you can break the bonds, and to be able to piece them all together. So, for example, this compound here is a saponin. It's actually a very big compound, right? You have your gluon type, which is the sterol, lanosterol portion, which is hydrophobic, and then you have your sugars attached. Okay, so how are these all attached in space? We figure out by doing manual interpretation of the spectra. So this was done largely by Zen, and it's kind of where we are right now, because if you look at the metabolome databases, underrepresented ang marine compounds. A lot of the metabolome databases actually are terrestrial compounds, and so we really have to do our part in populating the spectral databases, these molecular databases from compounds from the sea. Okay, so we did use open source public databases, but like I said, they're underrepresented. So we resorted into manual interpretation. So napapanaginipan ni Zen yung kanyang spectra, paano niya ika-cut yan. So you will know once you do this. But we persevered, they persevered, and they were able to generate the comprehensive metabolome. We're very happy because finally reviewer 4 accepted our paper. So plus one, it was this one was published in plus one just earlier this month, and so in a nutshell, we were able to identify over 100 compounds, okay, from the sea cucumber, and with half of those previously identified in other echinoderms. So okay lang, di ba? Kasi totoo naman they share similar compounds. But we were actually able to identify 25 new compounds because wala pang report. So ano kayang gagawin ng mga compounds na yon? So that's another question. And then so to make sense of this, we sorted them into 8 functional groups, right? So like I said, these are the saponins, yung actually para siyang detergent, di ba? Para yun yung ano niya. So it's anti-cancer, there's adjuvant activity, it's antifungal, and then we saw a lot of sulfated saponins, which are also have reported anti-cancer activities. And then you have alkyl sulfates and ceramides and sphingolipids, which are important for skin wound healing and tissue regeneration. And of course, your phospholipids and arachidonic acids, which are dietary PUFAs, etc. So we're kind of excited about this. Now, yung isang ginawa namin, we try to do the metabolome of the body wall and the viscera. Because what we learned from Ma'am Annette is that when the fisher folk get the sea cucumber and process it as trepang, a lot of times they just remove the gut and then throw it away. Okay? And then they actually dry the body wall. So sabi namin, okay, ano kayang meron doon sa body wall and sa viscera? And so in a nutshell, basically, the viscera is also very rich in bioactive compounds. And so we're trying to figure out, okay, baka naman wag na natin siyang itapon, di ba? Kasi we can harvest that, we can process that. And then, not one bit will go to waste. Kasi right now, they're just tossing this viscera, the gut. I mean, you can see, di ba, pag sa palengke, tinatapon lang yung innards ng mga fishy. So we can do something like that. And the interesting thing is, for this particular sea cucumber, yung body wall niya, pumunta sa saponins. Yung viscera niya wala. So merong mga classes of compounds that are actually just found in the body wall and some that are just found in the viscera. And later on, it's coming together, the story is coming together. But from the product development perspective, we're kind of happy because now we're like, okay, let's use the viscera because, they're just discarded. Okay. One of the things that's in the viscera is this polyunsaturated fatty acid, which is what we call arachidonic acid, right? And arachidonic acid is non-essential, so you have to eat it. And then it's actually important for wound healing and skin health. So marami ito sa sea cucumber, KS horrets. This is actually how we do the analysis. So just there's also a lot of sulfated compounds, and then a lot of the a lot of it is in the internal organs. Wala masyado sa body wall. So excited kami kasi okay, ano kaya pwede natin gamitin dito sa sulfated sterols na to? Okay, they're actually involved in inhibiting thrombin, yung sa and also antimicrobial activity. So there's a lot of leads there to be able to utilize this compounds from the viscera. Now this one is interesting. So we got the extracts and then we asked Dr. Mazaheri from IB to look at the assay. So this is a scratch assay. So essentially, ang idea lang niya, may cells ka, isa scratch mo. Tapos titignan mo kung paano siya mag-regenerate. And then we found out that mas mabilis yung regeneration kapag merong sea cucumber extract. And so could this be, something that would lead to cellular regeneration? We could do that. Of course, the antibacterial, cytotoxic, anti-proliferative, marami nang nakapag-aral niyan. But we're kind of interested in this particular aspect of the bioactivity of your S. horrens. Now, this one is also cute. I think in light of Jimmy's, where's is Jimmy here? In light of Jimmy's defense last, uh, kailan ba yun? Friday, diba? Meron siya, tinitignan niya yung feeding yung feed sa juveniles. So this, I don't know the scientific, it's pufferfish pa rin, cantigaster, tama ba ma'am? Anyways, so one of my students, what he did was to get the extract of the sea cucumber and then put it in food. So parang in-add niya yung extract dun sa food. Tapos sinignan niya kung kakainin ba nung fish yung food, diba? Tapos ang cute yung assay niya. Ngayon ko lang nakikita itong ganito pala yung assays for biology. So parang when it spits it out, diba? Kailangan kainin mo siya ng control. Tapos kailangan kainin mo yung control para masabi mo na ayaw talaga na yung food, deterrent talaga siya. So he did all these experiments and then in a nutshell, what he found out was that ayaw talaga ni fish yung extracts from the body wall. Pero si viscera, kinain niya lahat. In fact, inubos niya. Ayan. So there's really something in the body wall of S. horrens that is a deterrent for fish. Okay. And then we can also look at, with mass spec, we can also look at dynamic changes to the metabolome. One thing that we ask is, okay, paano kung sinugatan natin yung sea cucumber, anong compounds ang metabolites yung kanyang ma-produce? And then can we follow that as it heals? Or stress. So in this case, we have a control. This is the black one, that's the metabolome, like I told you. And then meron kami sinugatan. So yung spicules, kinat. And then then they did the metabolome profiling. And then 15 hours post-stress, kinuha ulit yung metabolome. So parang in this, it's a very preliminary data, pero makikita nyo na adaptive yung metabolome. So from 30-minute stress, talagang na-stress siya. Nag-iba talaga yung metabolome profile niya. But after 15 hours, it's sort of like goes back to the original metabolome. And we can further tease this out by figuring which compounds are actually very, very different. So in this case, we can see which features or which compounds were actually up, mataas yung concentration sa stressed, at saka yung mataas yung concentration after. So we can follow the dynamics, quote unquote, of a particular compound, in an organism. So madaming possibilities from the chemical ecology, biology side of things. Okay. Now, this one is very preliminary, but we're trying to now get the neuropeptidome of the sea cucumber. I mean, just because there's so many adaptations, right? So signaling naman lahat yan. So this is Aidan's thesis, preliminary data. Meron na siyang mga na-identify na neuropeptides from the central nerve ring of the sea cucumber, and these are the kinds of peptides that we have that he has identified. So I think the next part would be to validate whether they're really neuropeptides or not. Okay. All right. So do I have time? Pwede pa. Okay. So yung last story ko would be the collagen. Kasi nga, di ba, yung sea cucumber collagen, it's really 70%. I mean, yung body wall talaga is puro collagen. So, saan siya pwedeng gamitin? So, we, for Zhang and Kim worked on the biophysical and physical-chemical characterization of collagen. So, we were able to extract them, and then using mass spectrometry, we were able to say that the collagen is heterotypic, meaning there's many different kinds of collagen, 9, 11, yung iba-ibang klaseng collagen. And then we also did some BSc experiments to figure out how stable these guys are. But I think I'll just point you to this. So if you want, you can read the paper on ACS omega. Basically, meron silang collagen, tapos we analyzed, we imaged the collagen. So a lot, AFM, SEM, ano yung itsura ng collagen, diba? And then, what was interesting, malikot kasi si Kim, marami siyang gustong try. So what she did was, sige, hinit niya yung collagen biofilm. Tapos nakita niya na nagkakaroon ng differences in the properties as you heat up the collagen. So in this case, before TME, it was translucent and soft. And then when you heat it up, it becomes opaque and brittle. And then once it goes back down to, to normal temperatures, it regains its, softness and translucence. And then, pag sobrang taas yung temperature, obviously, madedegrade na si collagen. So, may mga ganitong activities, right? So, we're kind of excited because we want to actually exploit this for possible applications like heat stable films, packaging, etc. So, yun yung next goal namin. Okay? Ito yung, sayang, sorry, wala yung video. Hindi ko siya na, yung, remember, kasi cucumber, upon stimulus, either nagsosoften, nagsistiffen, di ba, depende dun sa stimulus nga. So, meron siya mutable collagenous tissue. And so, Kim and Alan did some experiments on, mechanical analysis. So, essentially, parang yung torture chamber. If you look at it, you clamp the tissue on one end, and then you see how far it goes, meron siyang force. So, this is artificial seawater. This is potassium supplemented seawater, and then ito calcium-free. So, it seems like that the activity, that the stretchiness of the tissue is actually affected by salts. So pag titignan mo, ito yung artificial seawater, talagang okay siya, masaya siya. Pero pag nilagyan mo yung, pag calcium free, para siyang bubblegum. So na-stretch talaga siya ng malaki. So they all started with the same size of tissue. But if you don't have calcium in your seawater, right? Nagiging parang bubblegum, di ba? Masyado siyang. So basically, this is just the beginning, but we think that with some more tweaking, we can actually modulate the stiffness of these MCTs as a function of ionic strengths. Okay, so that's what we did for our project. So thankfully, we just got good news. We were going to be funded for phase 2. For Stacopus. So the next thing for us now is now that we know what's there, the next thing for us will be to develop extraction and processing methods for these compounds, and then slowly see whether we can have biotechnological applications for such. And then, because my colleague Lilibet is into lead compounds for drug discovery, baka meron siyang gustong ibigay kila Zab, kila Zilge para maaral. So, because they're more into drug discovery. Okay. And so Outlook, we're just getting started. We started this project 2 years ago. I'm kind of excited. Even with the pandemic, what the students accomplished. So we're just getting started. There's over 100 cucumber species, 40 automorphily processed. So last week there was a sea cucumber symposium here, so that was kind of exciting. We also really like to have the early integration of natural product screening with biodiversity, and I think we're doing this now on a different, with some leads with Irene and Fred. Of course, we can't do it alone, diba? We need to train more people, and so if you like puzzles, right? Kung gusto niyo yung master, kung that is something that's interesting for you, we hold annual workshops on how to do this. So libre siya. So hopefully by July next year, gagawa namin ulit. we need more mass spectrometrists. We need more people who can do metabolomics and proteomics. Okay. Ito, kailangan talagang gawin. We need to create a spectral and molecule database, lalo na sa marine. And I think this is what I want to start and populate for MSI. Yung, at least yung, para for example, kung meron kayong sea cucumber and when you do mass spec, hindi na kayo magma-manual interpretation, you just access our database and you can identify at least those that have been identified. And then let's just worry about those that, we still have to do manual interpretation on. Okay. I I always think about how to transition our basic research outputs to products. And it's a big problem. Product siyang iniisip. Kasi parang think about the products from your marine, diba? The higher the compound purity, the higher the value. Diba? So you have your energy and feeds here and you have your drugs there. So syempre, pinakamataas return of investment, drugs, diba? But to be able to generate this, you need significant investment in terms of facility and technical skills. So we really have to push for more facilities, more significant investments in biotech companies, right? But in the meantime, maybe we can have something that is, of intermediate value. Kasi masyadong mahirap kung if we push for drugs, wala naman tayong infrastructure ecosystem. So I was thinking of maybe whether to have pilot processing labs or plants next to the aquaculture facilities. And let's actually ask the community that culture these seaweeds or sea cucumbers and help them, right? Teach them basic chemistry, how to extract, and then maybe they can have value added. Then they can add value to their products more than just raw material. Kasi ngayon raw material lang tayo, di ba? Okay. But again, the other thing also perhaps is that we should examine yung tech transfer ecosystem sa Pilipinas. Kailangan mabilis. Kasi para siya mabilis yung innovation mabilis yung sa business. And we can't do it all in the academy. We wear so many hats. But if we push for that and then possibly have a institutional, linkages with the private industries, baka mas maganda yung adaptation ng ating products. Okay? All right, so that's it. And thank you all for your attention. I want to thank past and current members of our lab, our collaborators, and funding from OVPA, from OVCRD, DOST, for our work. Thank you so much.
Thank you very much, Isa. I did promise you she's on a league of her own. Yeah, so questions from the floor. Okay, Ma'am Lau.
Hey, Ice, thank you for that. Tanong ko lang, actually, palang tatlo itong magkakadikit. How do you ensure, kunyari sa CQ Combo as an example, how do you ensure that one, siya talaga yung tinitest mo, hindi yung bacteria yung kasama niya?
Yeah.
Two, are you doing this on primary or secondary metabolites? And then three, how do you ensure that the raw material is not degraded? I mean, eventually, no? Kung nagkaroon to talaga ng like, sabi mo nga sa industry, how do you ensure that yung mga importante ay hindi ma-degrade before it gets processed?
Okay, so your first question, how do we ensure that it's actually the organism and not the symbiont? We have to do that pa. Kasi right now we're just collecting the body mass and the viscera. So yeah, I agree. There could be, metabolites from the symbiont, from the microbiome, right? So that will be next. Pero parang I think may isang study before that they looked at the viscera and look at the gut microbiome. Hindi rin siya ganun kadami. And then the second question is if it's secondary or primary, mostly secondary. Yeah. The ceramized sphingolipids, they're all secondary. Not so much primary. And then the third one is kung madegrade. Actually, madali siyang madegrade. Diba? Kasi parang pag hinawakan mo siya, tapos pag tinignan mo ng iba, malulusaw siya eh. So, talagang mabilis magtrabaho yung mga estudyante. Kasi they really want to make sure that it doesn't, degrade. Meron kaming solutions to ensure like, okay, after we cut, we put it here. That's like the solution we use to preserve, sort of. Pero makikita naman talaga kaagad kasi yung S-horrens in seconds, in less than a minute, nag-disintegrate talaga siya. So, obvious talaga kaagad na degraded na yung material. At least for the collagen.
Thank you. I acknowledge the question pa. Dr. Srigan.
Kung yung samples na ginamit mo, both for the shipworm and for the Stichopus, were they from the wild? Both of them?
Yung sticky cucumber po, sir, ano siya, wild.
Okay.
So, kukuha naman from Bolinao. Nagpapabuild kami sa fisher folk. Pero meron din kami mga ibang samples na cultured galing sa hatchery kay Ma'am Annette. Yung Campylobacter wild siya. Pero pagka siya yung nagko-culture eh.
Wala sa Mabini ba yan?
So we have several isolates from Mabini, and then we keep them. So Lilibet keeps them, and then we grow them. So these are wild.
Yeah. So yung kasunod na tanong doon, if you take samples elsewhere, both for the shipworm and for the Stichopus, or even try to rear them in a hatchery, do you think that you will have the same chemical profiles for both?
Probably there might be changes because when we were doing this in my past life, I was doing this with fungi, and if you culture the fungi in the lab, you get domesticated. So nawawala yung original compounds na. So over time, nagbabago talaga, sir. So for things for compounds, for example, for compounds that we want fungi to grow, to have, sometimes we challenge it with something else para mag-remember niya, I have to produce this. Kasi in the lab, di ba, domesticated, eat mashed potato, they're happy. They're not shy.
Kailangan i-stress sila.
Kailangan stress.
So mga estudyante para maging productive, kailangan i-stress. Ganun ba yan?
Konti lang. Huwag naman pagsobra kasi maliliso. So, bibait akong ano.
So if that is the case, then if you then take samples from different environments, then would it be likely that you will see different chemicals from those organisms?
Yeah.
And dapat is better if they are in a stressed environment?
I think it depends on the compound that you are yung target. So if the target is a compound that is elicited, if it's challenged, then yes. But for example, yung sphingolipids, these are whether, you stress them or not, they're there.
So pwede siyang direction ng study ng estudyante? Okay.
Naghahanap kami ng students. Ayan.
Thank you for that, Ando. Any other questions from the room? Questions? Okay, thank you. So questions from here, the ADR. Okay. Helen?
Now, just to keep the conversation going, you mentioned drugs as one of the ultimate objectives, but I would argue that for the people at large, there's something more valuable, and that's food. Do you think your lab is possibly going to consider food processing of the body wall, especially there's collagen? I say if you eat the collagen from the sea cucumber, will your complexion become nicer?
If you eat it, no. Well, definitely functional food, lalo na sa atin. I think there's a group in CHE, Home Economics, that actually, kasi it's the palatability, how they formulate the, maybe we can work with them. But yeah, definitely functional foods more than, parang mas mababa siguro yung barrier for adoption than the drugs. Yeah.
Early on, you said that uh, organisms that tend to protect themselves would tend to produce more chemicals. If you go to the wild and think of, say, corals, fire coral would be one of those that if you touch it, you immediately get a burning sensation. Um, why not fire corals then?
Yeah, pwede naman, sir. Kasi wala ako nag-drive eh. Kita yung example.
May counter suggestion.
Hmm.
Pag-aralan yung crown of thorns para matanggal siya sa wire. Diba?
Oh.
Kung may use siya, makakatulong din.
Pwede. Pwede nervous ako.
Okay. Question from Rene.
Before lang, minsan, ang ginagawa nila, electrification to actually improve the concentration of a compound. You elicit the response. So you actually increase the concentration. Anyways, yeah. We can do that in the lab synthetically or,
Actually, related dun sa sinabi nila. So suggestion ng crown of thorns. Para maubos na yung crown of thorns, di ba? Pero, okay. But that's the question. After you've identified useful compounds from something, does that actually increase the demand for those things? Or do you synthesize those compounds after, diba? Or what's the track record for these things? So it's both right, there's a conservation aspect to it as well, diba? If you find something useful from something that might not be, too many anymore, diba?
I think so I attended a seminar before. So in this case, what they were doing was to identify all the compounds that they can for a particular sample. Tapos after nun, meron ng AI na, okay, can you make a biosynthetic pathway to produce this? Or can you create a total chemical synthetic route to make this? So versus, harvesting and then. So I think for us, what would be good is yung valorization, yung viscera. If it's just getting chopped, right, might as well use it. So baka doon pa yung extract and purify versus, but if it's drugs, mas malamang siguro i-synthesize na lang siya kasi yung purity mahirap talaga.
There's a question from Dr. Ferreira on Zoom. Related to Dr. Siringan's questions, can the sulfated compounds, for example, give an indication of environmental conditions, sediment-water interface, where the sea cucumbers thrive?
Oh, actually, I'm not sure kasi when you add the sulfation is actually, I'm not sure if it's enzymatic or ano what drives sulfation of the sterols So I'm not sure if that's driven by the environment. Maybe we should look into that. Kasi yung sulfation is more for solubility of the compounds. When you sulfate a sterol, it's really more for increasing solubility, increasing permeability. But I'm not sure how that is linked to, in conditions in the environment. Sige, cha, usap tayo.
Sa flow through, so saan yung naiipit ang output? Saan yung pinakamatagal?
Analysis, sir.
And that analysis would be with the ICP-MS?
Mass spec. So after we get, so basically, madali na kapag na-develop na yung method So we just, ensure that we extract it and then it goes through the pipeline. But the problem now becomes I have so much data, I have so much spectra, and then how do they actually identify? So I think doon nagkakaroon ng bottleneck. Lalo na kasi bata, di ba? Kailangan pang mag-aral how to do it. But even if matagal ka nang metabolomics, mahirap pa rin, sir. Kasi nga, mas underrepresented yung ano. So it's in the analysis na at this point.
But I guess that's the exciting part. So you're discovering new compounds.
Yes. Mapapaniginipan mo talaga, sir, yung spectra. Mapaniginipan mo, nag-break to, ayun. Yun pala yung peak na yun.
So para bumilis yung part na yan, anong kailangan?
Algorithms, I guess, so, better algorithms to figure out yung fragmentation rules. So I think the general community naman is going towards that, in global community. They're now applying machine learning for metabolome identification.
And also the database that ISA is starting for MSI, the spectral and molecular database. That one will really help remove the bottleneck. Para kahit kaunti na lang yung managinip, at least meron kang babalikan na database. Pero mas maganda kung mas marami ang mananaginip, right? Do we have questions sa Zoom? Wala pa as of now. Questions here on the floor?
Thank you.
So snacks will be served after this. It will be right at the dito lang sa pagkalabas niyo dito sa pinto on my left. We can continue the discussions there. But before that, let us again thank Dr. Yu. And may I request everyone to please join us in front for a photo op. Maraming salamat, Aiza. So, the session today is the last of the MSI at 50 seminar series for this year. And we will start again January. Let's wait for further announcements. Maraming salamat. Photo op, please.